Hello dear children, welcome back and I'm Dr. Jisha Joe. In this video, we will be discussing some of the important questions from the chapter Genetics, some basic fundamentals which you can expect for the board exams. First, let us have a look at a few definition type questions. Some of the words that you can expect for definition: heredity, transmission of genetically based characteristics from parents to offspring. Variation, the small differences existing among individuals. Genetics, study of transmission of body features from parents to offspring and the laws relating to such transmission. Or it is a branch of biology that deals with heredity and variation. Character, any heritable feature is a character. Trait, the alternative forms of a character are called traits. Homologous chromosomes, a pair of corresponding chromosomes of the same size and shape, one from each parent. Autosomes, the first 22 pairs of chromosomes in humans which are identical in both males and females and determine bodily features. Sex chromosomes, the 23rd pair of chromosomes in humans which determine the sex of the individual. Gene, specific DNA segments of a chromosome which determine hereditary characters. Genome, the full complement of DNA of an organism. That is, it includes all the generic and intergeneric regions of the DNA of an organism. The entire complement of DNA is called as genome. Allele, alternative forms of a gene occupying the same position on homologous chromosomes and affecting the same characteristic but in different ways. Dominant allele, the superruling allele is called as the dominant allele. Recessive allele, the allele which in presence of the dominant allele remains suppressed. Homozygous dominant, condition in which both the members of the allelic pair are similar. Capital T or small t, capital T is the homozygous dominant condition. Heterozygous condition is a condition in which both the members of the allelic pair are dissimilar. Genotype, the set of genes present in the cells of an organism, or it is the genetic constitution of an organism. Phenotype, the observable characteristic which is genetically determined. Karyotype, the chart showing the artificial arrangement of chromosomes according to their size and shape. Sex-linked inheritance, the appearance of a trait which is due to the presence of an allele exclusively either on the X chromosome or on the Y chromosomes. X and Y are sex chromosomes. X-linked inheritance is the inheritance of a trait which is due to the presence of an allele exclusively on the X chromosome. The inheritance of color blindness, hemophilia, etc. are examples of X-linked inheritance. Pedigree chart is a chart showing the inheritance of a character using symbols. I shall discuss about the pedigree chart at the end of this video. Criss-cross inheritance is the inheritance of X-linked genes as in color blindness, from mother to son or from father to daughter. Monohybrid cross, a cross between two pure breeding varieties taking only one character at a time. Dihybrid cross is a cross between two pure breeding varieties taking two characters at a time. Filial generation is a generation obtained by the mating between two parents. Punnett square is a square in which the gametes of one parent are written along one side and those of the other parent written along the other side, and the possible genotypes and phenotypes of the offsprings written in the sub-squares. Mutation, sudden change in one or more genes or in the number or the structure of chromosomes. Mutagens, any agent that causes mutation is called as a mutagen. Example, radioactive radiation is an example of a mutagen. Genetic engineering, the technique in which the genetic constitution of an organism is altered by introducing new genes into its chromosomes. That is, by the introduction of new genes into the chromosomes of an organism, the genetic constitution of the organism is altered. This technique is called genetic engineering. Genetic counseling, that is, the counseling given to newly married couples or prospective parents regarding the possibility of any undesirable trait which their child might inherit. And both genetic engineering and genetic counseling are two modern applications of genetics. Next, we shall have a look at some of the words that can be asked for differentiation. First one, to differentiate between character and trait. Number two, autosomes and sex chromosomes. Gene and genome, the difference between gene and allele, dominant and recessive allele, homozygous and heterozygous condition, genotype and phenotype, monohybrid cross and dihybrid cross, F2 phenotypic ratio of a monohybrid cross and dihybrid cross. What is the difference between the F2 phenotypic ratio? Phenotypic ratio is the ratio of the phenotypes. So in the F2 generation, what is the ratio of the phenotypes? In a monohybrid cross, it is 3:1, and in a dihybrid cross, it is 9:3:3:1. Then pure strain and hybrid strain. What is the difference? Pure strain is a strain having two similar alleles for a particular character, that is the homozygous condition. Whereas hybrid strain is a strain having two dissimilar alleles for a particular character. That means it is in the heterozygous condition. So I hope that is clear. Then give reason type questions. Like begets like. It means that young ones look like their parents. What is the reason? Because of heredity. That is, because of the transmission of characters from parents to offsprings. Offsprings are never identical to the parents. What is the reason? That is because of the crossing over and the genetic recombination that is taking place during the formation of gametes, which leads to variation. Again, next question. Children of the same parents look different. The same answer. It is due to variation taking place. That is the small differences existing among individuals. And this, the cause of variation is crossing over and genetic recombination, which occurs during the formation of gametes. Lion and tiger have the same number of chromosomes, yet one is distinct from the other. The reason? Because it is not the chromosomes that determine character, it is the genes that are located on the chromosomes which determine the character. Hence, lion and tiger are distinct from the other. Sorry, one is distinct from the other. Hemophilia and color blindness are common in males than in females, because hemophilia and color blindness are due to recessive genes located on the X chromosomes. And since males have only one X chromosome, and if that gene is located on that particular X chromosome, he will exhibit the disease. But since females have two X chromosomes, for her to exhibit the disease, the gene for hemophilia or color blindness should be present on both the X chromosomes. If the gene is located only on one X chromosome, she is just a carrier. The sex of a child depends upon the type of sperm that fertilizes the egg, because all the eggs produced will have only the X chromosomes, whereas there are two types of sperms which are produced, half containing X chromosome and the other half containing Y chromosome. So if the egg get fertilized by a Y containing sperm, then the child will be a male, whereas if the egg gets fertilized by an X containing sperm, the child will be a female. Mendel selected garden pea as his experimental material. What are the reasons? First one, many varieties of the plants were available in alternative forms of a character. Second reason, the varieties available are in pure form, or they were true breeding. The third reason, pea plants are self-pollinated, but cross-pollination can also be done in pea plants. And pea plants have a very short lifespan, so it was easy to conduct experiments on pea plants. That is why he selected garden pea as his experimental material. Inheritance of X-linked genes is also called criss-cross inheritance. That is because it follows a special pattern of inheritance, that is from mother to son or from father to daughter. That is why it is called as criss-cross inheritance. Mutation alters the hereditary material. We know what is mutation. Mutation is the sudden change in the genes or in the number or structure of chromosomes. So if there is an alteration in the gene or in the number or structure of chromosomes, the hereditary material or the genetic material will be altered. So mutation alters the hereditary material. Now coming to structured type questions. First type is sex determination. From sex determination in humans, the flow chart can be given, and you that is to fill up the certain blanks that are given in the flow chart can be asked. That is to complete the flow chart. That is, blanks will be given at certain regions. Like for example, the region where gametes have to be written, then one or two offsprings, the next generation individuals, the genotype will be given, and you will be asked to fill up the other blanks. So such type of questions you can expect from sex determination in humans. Then next is sex-linked inheritance. From sex-linked inheritance also we have done, I think 2 examples we have done from sex-linked inheritance. Questions like that, those which we have done earlier can be asked. Then pedigree chart, we will discuss about pedigree chart. You don't have analysis of pedigree chart, but I'll just explain to you the pedigree chart in the next slide. Then monohybrid cross, we have done, I think, 3 crosses, 2 or 3 crosses we have done on monohybrid cross. Please practice doing with all the other characters also for both monohybrid and dihybrid crosses, so that in one question is for sure for the board exam, either from the monohybrid cross or from the dihybrid cross. Using Punnett square, practice doing the cross. It might be mostly 5-mark question. You can easily score 5 marks by practicing it using the Punnett square. Then state the laws of inheritance. All the 3 laws of inheritance study, and it might be asked for stating. Then experimental material and the 7 characters which Mendel selected along with their dominant and recessive traits. The experimental material that Mendel used for his experiments was the garden pea plant, the scientific name of which is Pisum sativum. You should know the scientific name. When writing the scientific name, the first name, that is Pisum, is the generic name, and the second name is the species name. Generic name or the genus name should always start with a capital letter, and the species name should start with a small letter, that is sativum. S should be small, Pisum, P should be big, capital letter. And both the terms should be separately underlined. That is how you write a scientific name. Then the 7 characters which he selected and their dominant and recessive traits you should know. Now next is what is a pedigree chart. So pedigree chart, as I told you in the definition, it is a chart showing the inheritance of a character using symbols. Symbols are used, as you can see in the figure, symbols are used to show the inheritance of a particular character. Now here, the circles that you see here represents the females. You can see a shaded circle and a hollow circle, that is, which is not shaded. So the circles, whether shaded or unshaded, represents the females, whereas the squares represent the males. Then the shaded circle or square represents an affected individual. Shaded circle or square represents an affected individual. That means if it is a shaded circle, it is an affected female, whereas a shaded square represents an affected male. Whereas as you see in the next line, next generation, you can see a circle which is hollow there, which is unshaded, which represents a normal individual. So an unshaded circle or an unshaded square represents a normal individual. Then that line which you see there connecting the 2 parents, the first generation, you can see both are shaded, father, and that is both the individuals are affected, and you can see a line connecting the parents. That line represents the mating between the 2 parents. And the second line, you can see 2 individuals there, the shaded square and a hollow circle. Those are the children of the parents. The children produced by the mating of the 2 parents. So the first line, that is the shaded circle and the shaded square, represents the first generation, whereas in the second generation, the children shown are the shaded square, which is an affected male, and the unshaded circle, which represents a normal female. So here the parents are both affected individuals, whereas the children, they have a son and a daughter, but the son is affected, whereas the daughter is a normal female. Then normally in a pedigree chart we represent the generations. I told you the first we can see two generations here. The first generation is the generation of the parents and the second generation is the generation of the children produced by the mating of the parents. So in a pedigree chart we represent the generations using Roman numerals. So that is the pedigree chart. So understanding the pedigree chart, circle represents female, square represents male, shaded circle or square represents the affected female or male, or diseased female or male. Hollow circle or unshaded circle or square represents the normal or unaffected female or male. Then the horizontal line between the parents represents mating. The Roman numerals depict the generation. So in this pedigree chart, we can see two generations. Now here I am taking an example of the inheritance of a condition called polydactyly. Polydactyly is a condition where a person has extra fingers or toes. If you count the number of fingers there, you can see it is 6. So this condition is called polydactyly. And this is a trait due to the presence of a dominant allele. So that means a polydactylic person will have either capital D or capital D small d. That is either in the homozygous dominant condition or in the heterozygous condition. That will be the genotype of a polydactylic person. So, polydactyly is a condition where the person has extra fingers or toes, and this trait is due to the presence of a dominant allele, and such a person will have either capital D or capital D small d as the genotype. Then this is a pedigree chart. The first example, the first pedigree chart which we have seen, the same thing I have taken here. It shows the inheritance of polydactyly. Here you can see the shaded circle and the square shows both the parents are affected. That is, they are both polydactylic. So both the parents are polydactylic and the genotype of such a polydactylic person should be either capital D small d or capital D. So, if the parents were both capital D, all the offsprings produced would also have been polydactylic with the genotype capital D small d. That means all the offsprings or all the children of such parents would be polydactylic. If the genotype of the parents were homozygous dominant, all the children produced would also have been homozygous dominant. But here in this pedigree chart, you can see that in the second generation, there is an unshaded circle which represents a normal female. That means she is a normal female. She has only 5 fingers. And for a normal female, the genotype would be small d. So from where does the child or the female gets that genotype? That is, both the parents should have the allele in them. Then only the child will get receive the small d from both the parents. So that means the genotypes of both the parents should be in the heterozygous condition. That is, both the father as well as the mother would have the genotype as capital D small d, not capital D. If the genotypes of the parents were capital D, then surely all the children they get will have or will exhibit polydactyly. But in this from this figure, we can see that one of the child, that is the daughter, she is a normal person, whereas the son shows polydactylic condition. That means both the parents have the genotype in the heterozygous condition. That is, they are capital D, they have capital D small d as their genotypes. So I hope pedigree chart is clear to you. Then coming to the applications of Mendel's laws, which I have left out while teaching a monohybrid and dihybrid cross. The first application, it is the Mendel's laws enables us to predict the frequency of new combinations in the progeny of hybrids. So if we have a knowledge of the Mendel's laws, it will help us to predict the number of individuals that can be produced in the progeny of hybrids. Then second application is, it is very useful. A knowledge of the laws of Mendel is useful for plant and animal breeders to produce better breeds of plants and animals. Third application is new types of plants with new combinations of useful characters can be produced by artificial hybridization. So breeders can artificially produce new plants by artificial hybridization with new combinations of useful characters. So children, with this we come to the end of this chapter. That is genetics, some basic fundamentals. In the next video, we shall start with the next unit, plant physiology, the first chapter of which is absorption by roots, the processes involved. So I hope the chapter genetics is clear to you. If you have any doubts or suggestions, please give your suggestions and doubts in the comment section. Thank you.